everyone, and welcome to today's webinar titled Getting to Results Faster, Automation in Miltenyi Biotec Imaging Workflows. This webinar is a part of the 6th Annual Laboratory Automation Virtual Event. I am Antonina Salcido of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by Miltenyi Biotec. For more information about Miltenyi Biotec, please go to miltenyibiotech.com. Now let's get started. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our new live chat feature during the presentation. You can find the live chat located at the right of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Help Desk button located at the bottom of your screen within the navigation bar or from the lobby. Or just use the Ask a Question box to let us know you need help. You will also notice that the speaker has provided a survey located to the right of the abstract. This survey will be available during both the live session and the on-demand period. In addition to asking questions, we encourage you to participate in 3 poll questions. To participate, simply click on the option that best describes your answer. We appreciate your participation. Our first polling question is, are you looking to expand your imaging capabilities? Select all that apply. Yes, I have batch high throughput imaging needs. Yes, I need to image large intact tissues. Yes, I'm looking to deeply characterize the cells within my biological system. That question again is, are you looking to expand your imaging capabilities? I'll give you a few moments to select your answers. Our next question is, does your current imaging protocol include any of the following? I am performing large cell analysis. I spend a large portion of my time validating IF antibodies. I spend a lot of time clearing tissues. That question again is, does your current imaging protocol include any of the following? Please take a few moments to select your answers. And our final question is, after watching this webinar, would you like to request more information? Yes, I'd like to schedule a call with a Miltenyi Biotec imaging expert. Yes, I'd like to receive a brochure on Miltenyi Biotec's imaging solutions. Or no, not at this time. And the question again is, would you like to request more information? Thank you so much for participating in those polling questions. I would like to now present today's speaker, Dr. Amy Jablonski, Technology and Application Specialist at Miltenyi Biotec. For a complete biography on our speaker, please visit the Biography tab at the left of your screen. Dr. Jablonski, you may now begin your presentation.
Thank you for the introduction and for answering those poll questions. So I'm really excited to talk about Miltenyi Biotec's imaging workflows. And really how we're really getting, working with researchers so that we can find a more efficient and reliable way for them to get their data out. Now, while this certainly concludes automation, we have a whole list of other features and tools built in to make the barrier to getting, imaging done lower and allow the researcher to really focus on what matters, and in this case, the data output. Certainly, there's all sorts of, animation that can be used to explore biological systems, whether it's the creation of certain models or staining of cells. These are all things that maybe you've already had experience with. Now, these, even are frequently used in a whole host of multi-omic approaches like I'm showing here, right, to really deeply understand biology. So definitely we've got, just an example of say, a tumor environment that we would want to understand. We could use genomics, metabolomics, or transcriptomics, or even where we're going to be talking and focusing most in is the proteomic space. And each of these has a slew of established and some emerging technologies that really are able to generate the data that you would need out of that. Now, in the world of proteomics, which is where I'm going to be spending most of the talk today, there are some really common techniques, that you've even used and probably even used some automation in, including that of the ELISAs and flow cytometry. So the really nice thing is about these techniques is that you can really increase the throughput and get a lot of information out in a short amount of time. Now, though, when we think about it in the imaging space, things like immunohistochemistry and immunofluorescence, automation is just really getting going here. However, if you think about it, imaging, based spatial technologies have really emerged as a exceptionally powerful and versatile tool for assessing relevant spatial characteristics in biology. So we'll jump in and talk a little bit more about that throughout the talk. So, this really includes the whole gamut of ranges of biological scales here. We definitely could be talking on something on the, larger scale, like a whole mouse, being able to understand the spatial information of certain organs, where certain tumors are in that mouse. We definitely can then move into something a little bit smaller. So we could do, imaging of a mouse heart, right? Understanding the relationships of what's going on in that. This could even be smaller, and a lot of you maybe even have worked with creating organoids as model systems and being able to image kind of what makes up this and use it as a model system for certain biology. But then we can even go all the way down to the single cell level, as we've got here an example of looking at a a tertiary lymphoid structure stained with a lot of different markers to be able to understand kind of the characteristics of what makes that up and their interaction with all of those as well. So thus here at Miltenyi, we really recognize the need or the range of imaging solutions to tackle all these type of biological questions that may be able to come out of this, understanding spatial interactions in the biology across, all of these different scales from the macroscopic, with our large animals down to the single cell or microscopic level as well. And I like to really refer to them as solutions because it's not just the instrumentation. Obviously, I've got some images and we'll be talking about the automation and kind of the efficiency in these instrumentations. However, it really is the whole workflow, like we said, to be able to get to that data output even faster. So, I'm going to be focusing on two aspects of that. I'm going to be focusing on both the light sheet microscopy and then also the high content side. And so I'm going to actually start here with the light sheet fluorescence microscopy. Now, some of you may have already used this particular technique in different platforms. However, I'd like to kind of just spend a few minutes explaining what goes on in this particular technique so that we can appreciate where we put in the automation later on. So, for example, I am just using this zebrafish in the center of the screen, labeled with different fluorophores, so the little stars inside there. And in light sheet microscopy, right, the excitation and the detection paths are actually uncoupled and oriented perpendicular to each other. So this is represented by the two objectives or the purple outlines on your screen. So the excitation beam actually illuminates the sample from the side and the detection of the fluorescence. So now we've got a couple of the little stars inside of the zebrafish lighting up, that these are actually detected on the vertical axis. So we saw that that emission path now is on that lower portion of the zebrafish. So the really nice thing about this is that the excitation beam can propagate through the width of the sample, and therefore the light sheet itself is creating the optical section compared to some other technologies that you may be familiar with. This means that there's no requirement for blocking out of focus light, so thus you get a little more efficiency here, and on top of that, we have no amplification of the signal with certain PMTs. Instead, light sheet microscopes are really able to acquire that entire field of view in a single camera frame. And then if you combine this with being able to do the axial translation, so this would be, either moving the sample through the beam or moving the beam through the sample, this is able to then really quickly generate a 3D volume of the biology that you're imaging. And thus, when you combine this with tissue clearing, right, light sheet microscopy allows the imaging of a large range of sample sizes. So certainly, if we look on the screen, right, we've kind of got our range of what we would be looking at, and then some different imaging technologies listed below. Now, certainly, there are ones that are better suited for those that are smaller. However, if you want to be able to start moving closer to that larger range of, say, rodent organs and small animals, right? This is where we really can then, get past that wall that I'm showing with certain things like multiphoton and confocal to be able to get past that, right? And overcome these certain challenges that they may get in order to be able to get into those. So not only are you imaging, small samples such as Drosophila brains or organoids, like we showed you an example, right, that are really, really common in confocal workflows, we can then be able to get past that and image all the way to the organs and the small animals where really confocal usability has been decreased. So this brings in advantages, a series of advantages to the light sheet microscopy, including the ones that you see on your screen. First, really gentle with minimal folded bleaching, right? Because we have the excitation beam propagating the width of the sample, we're able to use that to create the optical section of interest. And so we don't have to raster and have a lot of light going to it. Second, as you saw on the previous slide, when we combine light sheet microscopy, long working distance objectives, and tissue clearing, this allows for a wide range of samples that otherwise would be, need to be optically sectioned to be able to investigate. And then not only that, we've really got fast acquisition time. So because we're not having that requirement of blocking out of focus light with a pinhole or scanning across a field of view to acquire the signal with PMTs, the light microscope really can achieve these faster acquisition times and still be able to resolve individual cells. So this has made light sheet microscopy a method used across various fields of science, including that of neuroscience and immuno-oncology, to really visualize these larger 3D samples, organs, in small animals, as you can see in the covers displayed on the screen here. Now, those of you that have done light sheet microscopy know that there is a lot more to just imaging the samples and being able to produce images like this. There's to achieve these types of cover art, there are a couple of considerations in sample preparation and imaging conditions. And this is where Miltenyi Biotec takes really a holistic approach, right? We're talking about being able to really get these, conditions done and optimized efficiently so that you can get to the point of imaging and looking at the data. And so, we're really trying to decrease that barrier so that researchers can focus on the data output and their scientific outcomes. So to begin, we've got the biggest challenge that people usually face is when staining thick sections or entire organs, it's really the complete penetration or diffusion of the antibodies through the entire sample. So to ease this burden, we have 3D IF validated antibodies that have been screened for functionality and optimized for tissue penetration. Now, you can observe this in the image on the right of a cerebral organoid stained with beta-3 tubulin. In the case of the Miltenyi Biotec 3D-IF antibody, the antibody has penetrated deeper, reaching the center of the organoid so that distinct features can be seen, whereas if you compare that to the image on the left side, we can see that there's not as much penetration. This impairs the actual imaging that we could be done in this. Also, these are recombinantly engineered reaffinity antibodies. They're directly conjugated to bright and photostable VO dyes. So this offers a lot of advantages of direct immunofluorescent staining, including reducing the staining time, great signal to background ratio, and then also minimal background signals as compared to indirect staining. The next consideration is the tissue clearing that you heard me talk about before. In normal live or fixed tissue, different materials within the tissue, including things like water, proteins, and lipids, impair the light passage. So you can see this kind of in the middle, kind of the schematic of a simplified view of tissue and all the materials that would be kind of causing things like reflection, refraction, scattering, and absorption. And this really decreases the light penetration, detection, and ultimately the image quality. So that's why you get something like the image on the right of this brain, and it's very opaque. So, the goal of tissue clearing is to render the tissue transparent to reduce these impairments for better imaging. So specific protocols have been developed using the MACS clearing kit for easy, fast, and effective immunostaining and support of different tissues. However, the system is not limited only to this kit. That has the flexibility and support to handle different kinds of clearing methods as well that you may already be working with. And last is the instrumentation to visualize the actual samples. So in combination with our cutting edge light sheet illumination technology and objective lenses to ensure this image quality across all the scales, this to be able to create the 3D volume of your sample. Now, there are a lot of different unique features and automation that's been built in with this that I'm going to talk about in the next couple of slides that really, I think, lend itself to the ease of use, flexibility, and quality of imaging. So we're going to go ahead and start with the actual light sheet optics themselves. So the ultra microscope is actually equipped with 6 light sheets, 3 on the left and 3 on the right. So if you think about this, if you only have one light sheet going through a very, even a cleared tissue, you might get something like the image on the bottom right-hand corner. However, by adding these different light sheets, from the left and the right, you get a much more homogeneous illumination, better sectioning, and then even better image quality. The other thing is that the system has 3 long working distance lenses to reside on the scope. There's no need for the researcher to really kind of decide which one would be best for all the samples that they may want to image. This also has a lens exchanger for applying demagnification and post-magnification capabilities of 0.6x up to 30x to really give you the versatility of each of those objectives on the system as well. Additionally, these objectives are fully automated. You just simply go ahead and you can, at a touch of a button, you can change them to the one you want and then automatically find the sample so that you're really streamlining the imaging process. And then last but not least is the large automated sample chamber for easy access and mounting of multiple samples, which we're going to take a look at in the next slide. So let's go ahead and let's watch this quick movie. So basically we're able to simply release and then mount samples into the imaging chamber like you saw at the beginning of the movie. So this could be a whole mouse or a series of organs. And now what you see is that we're actually, once you've loaded it in, right, you've got this batch acquisition that can be set up to automate the acquisition. As you're seeing us go through each of these different types of mouse organisms or organs, so that really, again, you're able to set all this up ahead of time, get these images done, and then in the end, just take that data off. So in the next couple slides, I just want to show you a little bit of examples of what can be, imaged with this system, right? To be able to get an idea of like the image quality and the types of data that you could get out of it. So we're going to first start with with looking at something on a scale of a couple of microns. So this is a brain organoid in this example. We can see Ki67 here in yellow, followed by the SOX2 in magenta, and the neuronal marker in beta-3 tubulin in cyan. In the next example, we're going to go ahead and actually look at something a little bit larger. Now we're going to be looking at a whole mouse heart. So kind of the context of this is if you're thinking about restoration of perfusion after acute myocardial infarction, this is the most frequent and effective medical treatment. However, during this process, it may actually inflict an ischemia reperfusion injury or IR injury, which you'll see as the movie is going through. And that the hallmark of this injury is loss of vasculature and extensive inflammatory response. So now as this movie plays through, we're going to be able to see using 3D analysis really allowed the researcher to simultaneously characterize the extent and localization of the damage and ensuing inflammatory responses in the whole heart. So that's what's kind of being rendered here. And the other thing is that they could look at acute and late vascular IR damage zones. And these had really highly localized and spatially structured neutrophil infiltrates that were, identified with the LY6G positive spots or the cells that you saw in the video. So moving along to now, the lobe of a mouse lung. So in this example, they were looking at human innate lymphoid cells and in the context of mucosal inflammation. So in the video that you're going to see, this is actually a 3D animation of reconstructed lung lobes after 24 hours after the IV transfer of labeled human ILC2 into the mouse with induced lung inflammation. As we see these rendered through, you'll also see that there are these red spots. Those are actually the accumulated human ILC2s inside of this mouse lung here that they were able to then quantify and kind of get a better picture of what was going on within this system. And the last example is actually a very interesting one. Again, this is looking at a whole mouse body. So, underneath the kind of the little icon of a mouse, you'll see that there was indeed a full mouse that was cleared, and that's what it looks like. Now, what we're doing in here is like the researchers are looking for a very reliable detection of disseminated tumor cells and the biodistribution of certain tumor targeting therapeutic antibodies within this entire body. So the idea being is that being able to see the entire mouse and all the different biodistribution of the cancer metastasis, this allows them to better understand what's going on with their system. So as we fly through the mouse here, you'll see kind of in gray just the regular autofluorescence. The blue is each of the organs. And then in the magenta that you saw, these are the different, the metastases of the different cancer. And so what you're seeing now is like a zoom in of some of these micrometastases of various sizes throughout the entire mouse. And that's what they're trying to target with the therapeutic antibody treatment. So now as we're looking at it, you'll see the targeted tumors are actually in white. The untargeted tumors are in magenta. And we can see that there is a difference in, there are some that obviously have been targeted, but there are still some that are not targeted throughout the mouse. And the one thing that they were interested in is, why is that? And so what they had to do in this particular case is that they excised these micrometastases, they sectioned them, and then used confocal to kind of look at the sizes and the co-clustering with some of the different markers that they were able to look at But the conclusion that they had was that the existence of certain factors in the tumor microenvironment were really influencing the efficiency of the antibody drug targeting. So that's not too surprising when you start to consider all that goes into the tumor microenvironment. While the tumor cells are definitely a key constituency of the tissue masses, they're also surrounded by a complex microenvironment that comprises of a mixture of stromal cells, immune cells, cytokines, and extracellular proteins. So each of these populations, if we're looking at a top level of these different immune cells, kind of in the orange boxes, the idea is that it's not just these particular cells, that underneath all of these, there's a subpopulation or subtypes within each one of these as well, in different abundances and also spatial locations that make up the tumor microenvironment. And as we saw in the previous example, right, each of these cell populations really can have differences across the macroscopic and the microscopic scale of these tissues and contribute to the unpredictability of some cancer treatments. And so that's really kind of where we came in, thinking that there's a need for appropriate technologies and techniques to understand the multiple facets of this tumor microenvironment. So, there's really been a push to better characterize the tumor microenvironment. There's been lots of methods that have been used to go across, the number of markers that you may need to be able to classify that. So in this case, there are things like flow cytometry has been used, a very high throughput, efficient way of being able to do fast analysis of single cells on a very high marker kind of context. However, by doing flow cytometry, you normally have to do something like dissociation, right, to be able to get those individual cells, to be able to analyze. And this loses all the spatial information that you would have gotten, had you kept the tumor intact. And as we saw, there is also the spatial distribution and the interaction between these that we want to be able to investigate, as they might play a role in what's going on in this case as well. And that's where really, the imaging side, or some, very common immunohistochemical methods have played an important role, in that they keep the spatial distribution or the spatial makeup of the tissue. However, compared to what you can experience in flow, is that up to this point, it had been very limited in the number of channels or the markers that you could use. And on top of that, if you were doing multiplexing in the immunohistochemical, traditional space, this would require not only serial sectioning, but also a lot of, hands-on time of being able to do all of these subsequent stainings. And so that's where really, we introduced the MACSima Imaging Cyclic Staining, or MIX for short. So you'll hear me refer to this later in the talk. But that basically we are going to be able to do all of this staining, right? Be able to get past the number of like the 4 or 6 markers that we were seeing with kind of the traditional approaches, but in a very hands-off way to be able to get past that number and really get this done quickly. So in the mixed technology, we have just a little like icon of our particular instrument there. We basically put in prepared tissue that has no markers on it. The first step is to stain with up to 3 fluorescent antibodies. So I've just got an example of, say, CD326, CD31, and CD4 is the first go around. The next step in this would be able to sequentially image these fluorescent antibodies along with DAPI, and then do the last step, which is erase that signal and basically prep the sample to be able to have the next round of antibodies applied. So we can see, we'll go for another round. We've got CD45RO, we've got collagen 4, we've got some other markers in here as well. We do the subsequent, the imaging of those, and then we're able to do an erase step again. So you can see that basically we will continue building this up for the number of markers that we need in this cyclic kind of approach. And, you go until the number of antibodies that you needed to reach for your particular question. Now, our record is 400. However, we, do a lot up to hundreds of markers because that's, what we need to be able to define the system that we're working with. And so if we think about it, there's a lot of advantages to doing it in this method versus, say, the methods we talked about at the beginning here. So we definitely can get hundreds of markers on a single piece of tissue. So we're not having to do a lot of serial sectioning, which can be very, is not always possible when you are working with rare tissue. Additionally, unlike flow cytometry, where you're dissociating the tissue to be able to then put it into the flow cytometer, you'd be able to identify each of those cells. We keep the spatial distribution of all those single cells. So now we're really able to start looking at those spatial interactions that we might have lost. And then on top of that, this is a non-destructive process. So in theory, once you've been able to do this, you could use it for other downstream technologies on, again, that rare piece of tissue. And so here's just another example of just kind of what we can achieve with the mixed technology. Definitely, we've got a couple of different publications, a nice one looking at both the proteomic, but also the transcriptomic space and correlating those. And then also some in the immunotherapy space. And at the very end of this talk, we'll actually dive into how all of these things come together to be able to do that last one on the right, looking at pancreatic adenocarcinoma and CAR T therapy. So again, being able to bring this all together for the researchers so that they can worry more about the actual data output, we've come up with this solution of, what I call 4 pillars to be able to get this done, in a rapid and efficient way. First is definitely the fully automated instrument. So this is all built together to be fully automated for the sample processing and the imaging. So this includes, having multiple spots to be able to put the number of samples that you want to image in there. Also to have the reagent plate in here. And this is the only place that basically the researcher would be interacting with because everything else is done on board. So it comes with a integrated liquid handling system. So this does all the staining, all the washing. The researcher doesn't have to actually interact with the instrument to be able to take the sample out, stain, and then wash and put it back in. Again, this is done all on board here. The other thing is there's a full integrated microscope as well that is smart in the sense that it has, as you can see in the bottom image, under the microscope, we've got 3 objectives built into the instrument itself. However, the instrument knows whether or not to employ the 2x, whether doing an overview scan or to use one of the 20x's based on the type of sample holder that you're using, whether looking at adherent cells or imaging through a slide. And additionally has the capability of keeping the sample in focus the whole entire time as well. Again, the idea being is that you just load your samples and the antibodies, press play, and then get on with your day. Because everything, again, is done inside the instrument. And then again, once you have that, the data and the images are taken, you can then go ahead and work with that data, which we'll actually see later. We can use automated approaches to make this even more streamlined. The other thing being is that giving you the capability of doing different types of samples as well. Obviously, we talked about tissue and how, wanting to work with maybe a couple of tissue slides that you have. We have this validated for multiple fixation types. So we FFPE, PFA, as well as acetone. But again, not everyone may be working with tissue and actually wants to do multiplexing on tissue. They may want to do something with adherent cells, which is where our Maxwell 24 imaging plates come in. So you could just culture your cells in there, fix those, and then be able to do the multiplexing in those. And then eventually we're also going to be able to do suspension cells. So if those that are working with those suspension cells, you can either cytospin them into like that Maxwell 24, or we can then have these little micro chambers where one individual cell would fall into each one of these chambers, and you can do the multiplexing there. Again, being able to run the gamut of what you may want to actually multiplex and get a better understanding in terms of the markers that are expressed on those cells. Next, the other thing we do in terms of trying to increase the efficiency of this multiplexing space, and again, lowering that barrier of people working with this type of technology, is we have a list of validated antibodies. So these come from our high quality recombinant antibodies. So anyone that has worked maybe with the reaffinity antibodies before know that they're really unique in that they are this recombinant technology. They give lots of consistency. They're highly reproducible. And the other thing being is that they're actually engineered with a mutated FCR region. So this minimizes the background that you're getting from that, and also eliminating the need for use of, say, an FCR blocking buffer. The other thing being is, like, again, these are all within our particular portfolio. You'll see that we also know that they're erasable for this particular technique. And the nice thing is while our portfolio may have, thousands and thousands of antibodies, it's not always that we have them, right, but that they also work with the technology. And that's where the second portion, or the middle column, talking about the extensive mixed validated antibodies. So we have done a lot of work in-house to be able to take the antibodies that are within our portfolio and test them actually with the mixed technology. So making sure that the staining at room temperature works, right, at a certain concentration, and that these would then get the designation of a mixed validated antibody. So the nice thing here is that, of all those, we've actually validated up to 500 antigens across both the human and the mouse. We've got different clones for each of these. And that when you also take into consideration the 3 different fluorophores that we use within the technology, like we talked about at the beginning of the staining with the up to 3 antibodies, that we're talking about around 2,000 conjugates already validated, so that you can just, pull them off the shelf and be able to use, again, increasing that efficiency of being able to ramp up and then also scale up your imaging and your particular panels. The other thing being is that we've also have this in a couple of optimized antibody formats. We certainly have the vials, so that you can kind of have the flexibility of going from, say, a couple of antibodies, 8 or 12, right up to then the hundreds that we were talking about, and really test those out and have the flexibility of doing what you want with the vials, and which is really nice. However, we also have these RIA screen plates that are more standardized. Again, talking about the efficiency and being able to do this in a very reproducible manner as well. We have the RIA screen plates. These are, again, the Miltenyi antibodies, but they are dried down into these 96-well plates that you see here, and then heat foil sealed. And all you have to do is, again, put the actual sample in there, and then put this plate in here, and everything else is done on board. So talking about really ramping up of, again, just churning through samples. These are a nice standardized way of being able to have also your antibody and get done faster. So this comes in a RIA Screen Max, so all the antibodies that are validated, or custom plates once you have a panel that you want to run through for every single sample that you may come across. And so while all that's great, right, we're talking a lot about the efficiency of being able to, get the imaging done. Again, everything done in the instrument. Now it turns into, what do you do with the data afterwards, right? And traditionally, this has been kind of the bottleneck, in the sense that, a lot of automation has gone into the imaging portion, but now you need to be able to identify and quantify and do things with the actual data output. And I think that's really a unique feature of being able to have the MaxIQ View analysis software along with this package. So certainly it is a great viewer. All those images that you took and all the layers, you can see those together. However, the real power in this becomes the automated detection of those single cells, right? So we talked about, being able to get that single cell identification, kind of like you get with flow, but now we want it with the spatial context. And that's what we're doing here. So for those of you that are not familiar, traditionally this has been done in a very manual way. You could either, go ahead and circle each one of the individual nuclei and say that's a cell, or use some other approaches based on thresholding to kind of say where a nuclei is or not. However, the really nice thing about the MaxIQ view analysis software is that not only we're doing the traditional thresholding and histogram that you may have used in the past, but we're also leveraging all the different markers that you have in this that you were able to image in the system as well, to really help and get better segmentation and be able to get that better cell segmentation out of it. On top of that, once you're able to identify those individual cells, like we see on the picture on the right, we're also able to extract all of these features. Again, there's no math that we have to do. The instrument is going, or the software is going to go ahead, identify those, and then spit out those features, so you don't have to think about what you need until like downstream of what are the different features or the different measures that I want to compare for my particular system. But it doesn't stop there, because again, we may want to be able to do some automated classification, right? Particularly in this space, maybe I want to identify certain T cell populations. And that's where this classification within the software also becomes a vital tool to be able to, again, get to that automated identification, so that we can quantify and look at the spatial relationships of those. And last but not least is also giving you the capability of being able to do some higher level, graphing or looking at things like a heat map for comparison, so that you can do all of this inside of the software, while also always maintaining that back to the individual image itself. So let's think about the different, automated things that we can get out of extracting the information of that particular biological system. We definitely can first quantify cells. This is a very common thing that people will do in this kind of image analysis. So I'm giving you just an example. We were looking at this particular ovarian cell line, looking at it under control situation and then a treatment. And what was very obviously, just like looking at it, is that there was a big change in the phenotypic, what was happening in the system of those that remained after treatment were much larger cells and also had a lot of morphological changes in these. And so the nice thing is that within this software, we were able to actually map and understand, how big these cells were, what kind of the changes in the morphology of the nuclei. But then again, coupling that to the being able to do the multiplexing, we were also able to start looking into what makes these cells unique. And that's where this slide comes in. We're looking at the control on the left and the treatment on the right. We've just got a couple of markers out of this particular study, looking at DAPI, just again identifying the nuclei, CD44 for a kind of the ovarian cell cancer line marker. But then, we've got these markers X and Y that obviously had a very different expression in the control and the treatment groups. And it wasn't just that we could see that there were differences. Again, within the software, we could actually get the software to extract on a single cell level, right, all the different intensity differences that we were actually visually seeing with our eye. And that's what these are. So marker X on the left-hand side, marker Y on the right-hand side. And again, each of those dots is an individual cell, right, for that different expression levels on the y-axis of the graph. And again, now we're able to not only visually see what's going on, but also quantify and actually put numbers to that change that we were seeing visually. Not only that, we can definitely deep phenotype. So again, for those that are, maybe not familiar, right, there's going to be a lot, like we talked about, in this tumor microenvironment that we would want to be able to understand. And just using the T cell immune profiling and tonsil is just an example of how deep we can actually get in being able to look at all of these different T cell subsets. So on the left-hand side here, I've just kind of, listed out how we think about this. Definitely CD3 is a particularly good T cell marker that everyone looks at. But again, we were looking at these subsets of being able to get down to things like T helper cells, cytotoxic T cells, even things like memory T helper cells. And you can see kind of the markers that we were looking for in each one of those classifications. So the other nice thing, again, is that within the software, I can kind of make a visual gating strategy, I'll call it, right, inside of the software, so that no matter what sample I put in now, right, I can say this is the same kind of phenotype that I'm going to be looking at for each one of those. And so as an example, we just look at that first top level of CD3 and CD45 for the T cells and then kind of breaking them up. So we can again see that on the actual image here. In green, we've got our CD3 positive T cells. We can then see those particular T cells broken up into T helper and cytotoxic T cells. So now all these cells have again been classified into those two particular populations. But the other nice thing is, again, because we have all those markers, we're able to even go down to this very bottom level, which a lot of times is not looked at just for the lack of having those number of markers in a particular panel. And I think the other really nice thing is again, we can definitely do this. We can visualize it on the image, but we can also get numbers out. So we've got, the quantification again of the number of cells, the percentages of their population. This is all generated automatically in the software. And again, it's a nice thing to see that maybe these particular cells are for interest of your research, but there's a very small portion of the population, and this is definitely what we want to be driving towards. And last but not least is being able to spatially map all of these different things, right? That's why we're keeping the tissue intact so we can understand these spatial interactions. So I'll show you kind of two examples, again, that the software can do. First is distance mapping. So we've got here just an example of some markers for breast cancer in this particular image. So these are cytokeratins, and then the blue for all of the DAPI. We do the same, I talked about doing the T cell gating strategy in the previous one. We can apply that same idea here to be able to see all the T cells, which are now being displayed on that same section, but in red. And then the other nice thing is, again, I can tell the software I want to be able to automatically make these zones and look at the different, something that's very hot or very close to the particular tumor. So this will be in red. To something, we've got a rainbow effect into the what I'll call further away or colder region of the particular T cells. On that one, I overlay it, right? White is now where my tumor was, and we can see the red all the way out to the blue. But on top of that, I can actually then automatically just engage a heat map to be able to see if there's any signatures in the differences of those particular zones. We can also do this by doing that same kind of gating strategy against other different types of cells, which I'm not going to go too much into. We actually have a nice webinar looking at the lung edema carcinoma and how this kind of all comes together. But the idea being is that once you gate each one of these particular cell subsets, you can then make the spatial relationship between the two of them as well, and be able to get a heat map that way. So just as a quick kind of introduction to that is that we've got the distance for each of the tumor, the T cells, the macrophages, and then also those particular populations across the bottom of this particular heat map. So you would imagine that the deepest color, the purple, is closest, and then the red is, or that deeper red color is the furthest away. So that makes sense why we've got a diagonal for each of these. And, just drawing some conclusions, we see that the T cells are actually further away than the macrophages from the particular tumor. And so again, I like to end with how all this comes together, being able to use all the markers that you can automatically image, but then also the software to automatically detect individual cells and make some conclusions out of it with this example of CAR T cell therapy in PDAC or pancreatic ductal edema carcinoma. So for this particular paper, it was part of the mixed technology was actually part of a larger workflow too of engaging a lot of automated devices, right, to really start identifying a particular target of interest so that when you get down to the generation, the CAR T, you have a few good targets to use. But I think the other nice thing is you can wrap this back around and actually look at, the mechanism of action in this particular case of why maybe a treatment worked or didn't work. So just a little introduction here. We've got, we started with screening a bunch of xenografts, so where we've got a lot of tissue available, right? We can actually use flow cytometry, but that once they wanted to work into primary PDAC or pieces of tissue, they didn't have as much. So that's why the mixed technology was very useful. So they spread that across the mix and the flow cytometry, then also looked at it in off-target effects in the healthy tissue, and again, did the CAR T generation and the actual validation. So here's just a quick image of, like, again, all of the different images that you can get out for the different markers that were actually investigated. But again, the really nice thing is that once you have those, you do the single cell identification, and then you can get a really nice heat map out for each one of the cells that you've got in all the markers. So for reference, going on down, so each of the rows is actually a individual marker, and across the top, each column is actually one cell. And what they were able to do is, again, identify regions where the markers of the particular markers for the PDAC gave a specific region, and then of course the immune cells, and they were looking for targets, that would be highly expressed in the tumor region of this heat map, but not in the immune cells. And so while it's really great to be able to have this, very quick and easy way of identification, I don't think I'd be remiss to not also say that it's great to have the images, because not only can you verify what you're seeing in the heat map, where you, like here, where we can, verify that CD59 was not tumor specific because it did not at least overlap with the green now for the cytokeratin, which would be our tumor, and then the marker of interest would be in red, and so this would be the CD59. And those that were tumor specific, so that's something like CD318, where you now see the green and the red overlap very well. But you can also kind of look if there was one that you were unsure about or was not sure about them, wanted to investigate the morphology. You can also do that, like in this last example with the CLA. So once they were able to identify a couple of targets, this was their top three. So CD66C, T-span8, and CD318. All that I'm showing here is that upon a tumor microarray, they were looking at the PDAC. So this was the actual tumor that they're looking for, but we're able to also put a bunch of healthy tissue in that case too, stain for those particular targets and see whether or not they were expressed on any healthy tissue, and that if they do generate the CAR T, would there be any kind of off-target effects that would lead to either decreased efficiency or, other dangerous effects. So what they were able to at least see is that CD66C and CD318 did not have a lot of off-target staining, whereas T-span8 did have some in the colon, but they were able to use this maybe for some other treatments as well. So they looked at that and generated that as well, which is the last portion of the actual CAR T generation administration. So on the left-hand side, we're actually looking at the mice and looking at the xenografts in there and the bioluminescence for each of those for the CD66C, T-span8, and CD318. The V underneath is just the different lengths of the linkers on the CAR T. And what we can see, at least visually, is that the CD66C and the CD318 was very good at being able to do the reduction of the actual of the size of the tumor. We can also get that graphically on the right-hand side. And what's really shocked here is that the difference between the CD66C and the 318, which were the top two targets, is that there's a marked difference between the actual reduction of the tumor size. And that's what led to being able to do this also in terms of the mixed technology of looking at what could possibly be going on in the system as well. So looking at the actual CAR T cell infiltration. So for reference, CD66C will be across the top row, and then CD318 is across the bottom row. And in the first couple, we were looking actually at the CAR T infiltration. So this is looking at some T cell markers, so CD4 and CD8, but then the specific CAR T being labeled in the blue of the LNGFR. Also making sure that there, whether or not there's actually tumor left in there, what kind of, macrophage infiltration we get. But also, since there was a question of a possible downregulation, they were also making sure that CD66C and 318, whether or not they're actually still expressed. And that's why the conclusion is from here was at least there wasn't a downregulation of the CD6C that was leading to the differences, but might be something that has to be investigated more. And so with that, I'm very happy to take any of your questions, and I hope that this was helpful in terms of seeing how bringing in automation into their whole entire imaging portfolio, it could be really useful for different applications that you're working with now.
Thank you, Dr. Jablonski, for your informative presentation. We will now start the Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. Now let's get started. Our first question is, can I take advantage of my own automated analysis pipeline in combination to the MACSIQ View software?
Yeah, so that's a great question. Thinking about the MACSIQ View software, I think there's several ways that you could combine this with existing analysis pipelines. First, there's user-generated segmentation masks people have obviously worked on. You can import these into the software to identify the cells and then use the, downstream tools that are built in. Definitely also there are features generated inside MACSIQ View that can be exported, and then, certain measures that you could then use that and put into your own statistical package. And then lastly, which I didn't touch on, but we actually have the capability of importing Python scripts that you may already be writing into the software. So it's combined all into one for a really complete workflow.
Great, thank you for that response. Our next question, how many samples can be run in the MACSima at one time?
Yeah, so the MACSima has a total of 8 slots in the sample and reagent chamber area that I alluded to earlier. It can hold up to 8 slides at a time. Typically, though, we run about 3 to 4 samples at a time based on our antibody panels, right? We're doing a lot of these so that the sample doesn't sit unprocessed for an excessive amount of time.
Thank you for that. Another few questions. How big of a sample or how many samples can be loaded into the Blaze?
Yeah, so I think I showed you guys, right, that it can go really small to even that really large, cleared mouse. So the Blaze has a large chamber sizes, which really can accommodate all of those without being able to need that sectioning. So our standard sample chamber is capable of actually mounting 5 cleared intact mouse organs to be able to do in one run. We also just released a new XXL chamber, which actually has the capability of fitting a whole human organ or an adult mouse. So we were showing you some more younger mice, but now we're going to have the capability to do that. Additionally, right, the mosaic and batch modes that are built into the software actually give you the capability to do this all and process all these tissues in a single run.
That's awesome. Thank you for that. All right, we're going to wrap with this last question. What type of clearing methods can be used with the Blaze?
Yeah, so I alluded to the Max Clearing kit, which obviously is a great way to get started. But I know that a lot of people that work in the kind of light sheet space already have really worked a lot on, optimizing their clearing methods for their particular sample that they're working with. And the Blaze is designed really to be compatible actually with most clearing techniques. So just to give you a little idea, each objective actually has a series of interchangeable dipping caps that are corrected for the range of refractive indexes and Abbe numbers that corresponds with most of these different imaging solutions that you may be working with. Not only that, we also have a refractive index adjustment collar that you can make minor changes to as well to match better to the refractive index of your imaging solution. But I think the really nice thing, talking about the automation built in, is that the Blaze is fully automated and makes these more minor or fine adjustments in the light sheet and the imaging optics to really correct for the spherical and chromatic aberrations that you might be seeing. So I think it's kind of the best of all worlds, right? You can use what you're used to, but then you can also take advantage of some of those features really to optimize that and be able to have the best of both worlds with that.
Thank you, Dr. Jablonski. Do you have any final comments for our audience?
I don't. Thank you for the opportunity, and I hope everyone found this informative.
Thank you so much again for your time today and your important research. Before we go, I'd like to just thank the audience for joining us today and for their interesting questions. I'd also like to thank our sponsor, Miltenyi Biotec, for sponsoring today's webinar. Questions we didn't have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 2 years until May 18th, 2024. LabRoots will alert you via email when it's available for replay. And we encourage you to share that email with any of your colleagues who may have missed today's live event. Until next time, goodbye.